All right. I was informed that half of chapter 17 is missing, so let's go through what we missed. So okay. First part, the four outcomes of an antigen antibody reaction. Okay. Okay. So the antibody complex formed by the antibodies by the antigens. Okay. The strength of the body is the affinity. Some antibodies have greater affinity for the antigen than others. And selecting for antigens with or antibodies with high affinity to COVID-19 is what researchers are trying to do right now. That's one of the things that they want to do to try to find a vaccine. Okay. Okay. The protects the host by these processes, and you can see these guys right here. So this is the glutination, this is the IgM, this is mostly IgM. So they show IgG, but this is actually done by IgM. IgM makes big, huge clusters of these things. Okay. Complement is done by IgG. Okay, now that answers one of the earlier questions. Complement is done by IgG. Okay. Optimization is done by both. Okay. Just having antibodies around attracts neutrophils and macrophages. Okay. And then the antibody-dependent cell-mediated cytotoxicity is how we actually kill worms. Okay. And then last but not least, neutralization. Okay. I told you guys have these videos like this came through, so I'm gonna skip that. Okay. Okay, so now we're talking about the other wing. Okay, first the humoral wing. Now we're talking the cellular wing. Okay, and this is managed by T cells. Okay, T cells mature in the thymus. Okay, where they undergo something called thymic selection. Okay, so the only thing that passes thymic selection is things that do not bind to things that are in you. Okay, that's called negative selection. And then the other thing is it doesn't pass the test if it binds things that are very weird shaped. That's positive selection. Okay. Um, okay. After that, if they pass, and very few things pass, okay, I think it's something like 1 or 2 out of 100 pass, okay, and then if the T cell passes, that means it's going to be useful somewhere, and so it's going to migrate from the thymus to the lymphoid tissue, mostly the lymph nodes, okay, and then attached via antigens via T cell receptors or TCRs, okay, okay, okay. Don't worry about M cells, higher patches, okay. Antigen presenting cells, okay. Antigen presenting cells have MHC2. Okay, MHC2 is a way to present antigen. Okay, so dendritic cells, macrophages, helper T cells, they all have MHC2. Okay, so this is a dendritic cell, macrophage. Okay, okay. So the T helper cells, what they bind to is the MHC2 on B cells and APCs and dendritic cells and stuff. Okay, these are going to be T helper cells. The T helper cells are also known as CD4 cells or CD4 plus cells. Okay, what they're going to do is start cytokine signaling. they will activate the B cell, or they will activate the macrophage, or they will activate other stuff. Okay, the cytotoxic T cells. Okay. Okay. So what happens is here. This is a T cell, T helper cell. The T helper cell, okay, will recognize the antigen right here. Okay, and this will then activate. Okay, it will send signals to activate this the self-activate here, and this cell, this cell will activate active B cells, CTL, cytotoxic T lymphocytes, and macrophages. So once you get a go signal, it will activate itself, it will clone itself, and then it will start activating B cells, cytotoxic T lymphocytes, and macrophages, all of which can bind to the same signal. Okay, that means there's a pathogen out there that I can kill. I have a job to do now, so I'm gonna proliferate myself into million little bits, and then I'm gonna activate cytotoxic T cells, which will kill individual cells, and B cells, which will make antibodies for the bloodstream, and then macrophages, because macrophages do their thing everywhere. Okay. Okay. You do not have to know the subclasses of Th1 and Th2. Okay. Okay. So T helper cells activate all these cells. Okay. Okay. You don't have to know about T regulatory cells. They usually suppress things. Okay. Cytotoxic T cells. Okay. So here is a virus infected cell. The virus infected cell will always present the antigen on the surface. Okay. So virus infected cells will always present what the virus is. Okay. If you're not infected with anything, you will present whatever the hell you ate, basically. And so no matter what, this happens. So viruses can't just hide there. Once it gets in, it will get presented. Okay. Cytokines. So these guys will activate. These guys will release. So this is a T helper cell. It will signal a cytotoxic T cell. When a T cell binds to the cell, it will activate porphyrins and granzymes. The porphyrin will poke holes, and if that's not enough to kill the cell, the granzyme actually causes apoptosis. Okay, this is apoptosis being caused here. The cell blebs. It so it can be broken off into little pieces that can be easily swallowed. The other important thing that it does, it destroys its DNA. Remember, the DNA is everything inside a cell, right? This is an infectious particle. It's always going to be DNA. So the first thing you want to do when you kill yourself is chop up all the DNA. So you make sure you chop up all the viral DNA along with it. Okay. 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 Natural killer cells. Um, we don't actually know exactly how they work. They kill a bunch of different stuff. Okay. They destroy cells that don't express MHC1, with the exception of red blood cells. Okay. I don't really know how they avoid that, but they do. Um, okay. They kill virus infected tumor cells. One of the ways that tumor cells try to avoid getting killed by the other parts of the immune system is to downregulate MHC class 1. So they don't show that very often. So you don't see that, and you don't know it's infected. Okay, and stuff like that. And natural killer cells get to those guys. Okay. Okay. Um, Antibody-mediated cell or antibody-dependent cell-mediated toxicity. We showed you guys a picture already, but it's basically this. Okay. Um, Antibodies are generated. Antibodies bind to the surface of the worm, but just having the antibodies is not enough to kill the worm because, like, macrophages can't swallow a worm. What they do though is they attract eosinophils to the end of the Y, and that causes them to release granzymes, lytic enzymes. They're actually called granzymes. They puncture. They cause apoptosis. Porphyrin enzymes cause pores. Porphyrin. Okay, porphyrin. Okay. Okay. Primary and secondary immune response. Okay, so hugely important thing right now. Okay, primary from secondary immune response. You can bet all the money that you have that this will show up on the test in some shape or form. Okay. Okay, the secondary immune response is also known as the memory response. Okay, it's the second exposure to an antigen. Okay, so here's what happens. Okay, one. Okay, I initially inject you with the antigen, the initial exposure. Okay, it takes about 10 days for the IgM response to build up, and it takes about 14 to 16 days for the IgG response to show up in the maximum. Usually we quote it as 14 days. Okay? Okay. So this takes a while. Okay, so you get IgM response first and then you get IgG. Okay? The second exposure after you've recovered, I inject you a second time. The time it takes to generate this response is about 3 days, okay, to get really high. This is actually kind of incorrect, but it keeps going up, which is true, okay? The total response is about 1,000-fold bigger, okay? And the other thing that you notice is that there's not much of an IgM response. It's been switched to IgG, okay? That's why IgG is the major immune thing. Okay, um, uh, IgM only works temporarily, so IgG spits out huge response. Okay, so you can see the exposure is much shorter. The later exposure to this is much shorter than the original, right? The original takes about 15 days here. Okay, this is about 5. They show about 5 here. In actuality, it's about shorter than that actually. Okay. Okay. That's why you want a vaccine, because you want to fight off the virus with the secondary immune response, not the primary. Okay. Okay. The 4 types of adaptive immunity. Okay. Okay. So naturally acquired is you get it from the environment somehow. Artificial required is injection of the thing into you. Okay. Now there's active immunity and passive immunity. Active immunity, okay, um, only occurs if your body is challenged. Okay, if they inject the antibody into you already, then you don't build up the response. So you don't have to do anything. So the immunity is passive. Okay, if uh, if you get the virus or get a bacteria inside you that's replicating and stuff like that, then that is active immunity because you're actively fighting it off. Okay. So here you can read all these. Okay. Okay, so make sure you understand these guys because it doesn't show up on the test. All right. So hopefully that explains everything. Um, you guys can all thank Shuka for making me do this. But she pushed me to make sure you guys get all this information. So say thank you when you see her.